guy doing?
Can you draw it down? He's so nice to us. He's so nice to us. Oh, it was very beautiful, very very. He wants to stay. He's very nice. He wants He's very nice. He's very very calm. I love him. He's so nice. He's so nice, so approachable. Really very good. And we have genetics.
Good morning, everyone. Welcome to another Monday. Where is everyone? It's like daylight savings time. Everyone's still asleep or something. This is an empty classroom. Wow. Which is kind of too bad because there's a test coming up on Friday. Anyway, so test 3, which you guys wrote last Friday, it's all marked. Everything looks good. The average was just under 79, which is about 4% higher than the previous one. I don't think there's any problematic questions. I'm trying to add the feedback because I didn't want to risk having anything released prematurely. So sometime tomorrow you'll get test 3, and a reminder, test 4, because we have to eliminate one of the tests, is this coming Friday. So this test, this upcoming Friday, is going to only cover 2 lectures. So this lecture today and then Wednesday's lecture will be covered on a test on Friday. Okay, it's not a lot of material. I'm still going to make it 20 questions. If I make it less questions, I think people will be upset that each question is worth a lot of value. So it'll be 20 questions just like the rest of them. It's only going to cover 2 lectures worth of material. Right? Today's lecture and Wednesday's lecture. There are now people who have missed more than 2 tests. So in the syllabus is a pretty good explanation of what happens if you miss more than 2 tests. There's built-in flexibility. There are 4 tests. The top 3 tests count. Right, so that flexibility means if you've done well in your first 3 tests, you don't even have to write test 4. Nothing happens. We use your top 3 tests. Because there's built-in flexibility, that means there's certain things that fall into place such that if you only miss 1 test, you don't write a makeup. Okay, so people who have missed 2 or more tests, there will be a makeup sometime the week of March 17th, depending on the availability of the students to write it. So that makeup is going to cover everything so far, right? Because if you've missed more than 2 tests, you've missed a variety of different topics. So the makeup is cumulative. It covers everything we've covered so far. So if you've missed more than 2 tests, and you provide some sort of documentation, it's all explained in the syllabus. If you miss 1 test, this happens, 2 tests, this happens, etc. Okay, but if you miss more than 2 tests, the makeup is next week, and it's cumulative. Alright, so let's get on to the exciting world of cell adhesion. Okay, so we're going to cover sort of 4 different types of cell adhesion today. Hopefully, it's quite straightforward. So there are 4 things we're going to talk about. These things called tight junctions, gap junctions, which provide adhesion, but they don't necessarily provide strength, and we'll talk about that in a second. And then there's cell-cell adhesion, right? So there's a number of ways cells can stick to each other, and also cell extracellular matrix adhesion, cell ECM adhesion. Okay, so when you look at cells, any cells in your body, they're stuck to each other, where multicellular things are held in place. So this example here are epithelial cells, and we'll talk a lot about epithelial cells, but in general, these sorts of adhesions are present in other cells as well. So when you look at epithelial cells are unique because they're sort of an apical and a basal surface, because the apical surface is always exposed to the outside. Right, so for your skin, your apical surface is literally the outside. In your intestines, the apical surface is in the lumen of your gut, which is the same as being outside. You just kind of put food in your mouth and it comes out the backside, and that whole alimentary tract is sort of like the outside. So apical basal polarity is crucial to epithelial cells, and we'll talk more about that when we're talking more about epithelial cells later. The point is, all of your cells have these different ways to stick together. Some of them provide sort of functional purposes, and some of them provide strength. We've already talked about some of these things before. We've talked about desmosomes and hemidesmosomes, right? And we'll talk more in depth about them now. So all cells have cell-cell adhesions. Different cells have different combinations of these things. So let's kind of look through them to see what they are. All right, starting off with sort of tight junctions and gap junctions. So tight junctions and gap junctions provide adhesion. They kind of glue cells together, but they don't necessarily provide strength. And the reason they don't provide strength is that they're not usually linked to the cytoskeleton. I've mentioned several times the plasma membrane is a weak structure. You know, phospholipid bilayers aren't strong. You need proteins for strength, and that's where things such as keratins and all these intermediate filaments and actin play. They play a role of giving strength to the membrane. So gap junctions and tight junctions have function in cell adhesion, but they're not necessarily linked to the cytoskeleton in a way to provide strength. It's not that the cytoskeleton isn't related to gap junctions and tight junctions, but the cytoskeleton does not provide strength through gap junctions or tight junctions. All right, lots of words there, sorry. So what are gap junctions? Gap junctions are open channels that exist between cells, so they link cell membranes together. So the easiest example is an epithelial cell. If you have one epithelial cell on the left, one epithelial cell on the right, gap junctions form an open channel. And that open channel allows small molecules to go through. Gap junctions are made up of proteins called connexins. I think humans have about 21 connexins, and they have different molecular weights. So you can read about connexin 40, connexin 43, connexin 46, connexin 30. There's different molecular weights of connexins. 6 connexins come together on one membrane to form something called a connexon. So you have 6 connexins on one side, one cell, and those connexins can all be the same family member or different family members. So they can all be connexin 43, or a mix of connexin 43 with connexin 30 and connexin 26, etc. So if you use different connexins, it gives different properties to this channel in terms of how big it is and what sort of things can pass through. Okay, so you have 6 connexins that make a connexon, right? And then you have a connexon on one cell and a connexon on another cell, and they come together to form a gap junction. So it's a total of 12 proteins. The dodecamer, I think, something like that, but I don't see that. Okay, so 12 proteins come together to form this gap junction. That's largely a pore, an opening between the two cells. They have different functions in terms of the sizes that they are, but for our purposes, they allow small molecules to pass through, certainly ions. The only thing that I will talk about or test you about between the things that pass through gap junctions are ions. So sodium, potassium, chlorine, calcium, they can pass through gap junctions. Other things can as well, but we're not going to deal with those because not all gap junctions can allow the passage of all things. Okay, so gap junctions allow connections between cells. They're literally an open channel between cells. And they allow communication between cells. And you'll see when we talk about epithelial cell function why that's important, because it allows cells to maintain equal amounts of ions across multiple sort of cells. So if you have a large layer of epithelial cells, they will all contain the same ions because ions can diffuse through these gap junctions. But because they're open, that's a bit of a danger, because if you cut yourself, if you cut your skin, you're damaging the cell. Suddenly you have these gap junctions, which are largely openings, they're actually channels on the plasma membrane, and the cytoplasm of your intact cell can literally leak out into the cell that you just damaged. So gap junctions are open channels. They allow they allow ions and things to flow through, but if you damage it, you don't want that flow to continue. So there's a number of ways you can regulate gap junctions. They can be closed, but the only one I want you to sort of memorize is that gap junctions are closed when they're exposed to extracellular calcium. If you remember, we already talked about this, there's almost no calcium in the cytoplasm. Right? But there is lots of calcium extracellularly. So if you cut yourself, right, if you cut a cell, suddenly there's going to be calcium present, and that calcium is going to interact with the connexins in this gap junctions and close that gap junction. So gap junctions close in response to calcium because there's normally no calcium present. I may have said calcium is transported to gap junctions, it isn't. Make sense? Extracellular calcium is going to close them because normally they're not exposed to extracellular calcium. So gap junctions allow communications. Things can move between cells connected by these gap junctions. And most easy to memorize is that ions can pass through between cells. So you have gap junctions which don't necessarily provide strength, and you have these things called tight junctions that, while they have a function as a barrier, also don't provide strength. So again, if you look at epithelial cells form a barrier. Epithelial cells, their job is to form a tight layer so that nothing gets through. So you don't want pathogens going between your cells. In your gut, you don't want material slipping between your cells. You want food to be selectively transported. So when you look at epithelial cells, they have an apical surface and a basal surface. And you do not want material to go between them. You do not want paracellular transfer of stuff. You want material to be transported through the cell. You want the cell to actively take glucose from one side and put it to the other side. You don't want material to go between the cells. That material is regulated. Cells to go across an epithelial cell membrane has to be regulated. So what you do is that you glue the apical surface of epithelial cells together with these proteins that form gap junctions. So there's proteins called occludin, claudin, and jam. Okay, so occludin, claudin, and jam are transmembrane proteins that form tight junctions. They literally glue the membranes of cells together, particularly epithelial cells, particularly close to the apical surface of epithelial cells. So what they're trying to show you in the bottom right here is you have two epithelial cells, one on the left side and one on the right side. You can see these epithelial cells have microvilli, and microvilli are used to increase surface area. And so in between the two cells, and these are the plasma membranes, you have tight junctions that literally glue the plasma membranes together so no material can pass between the cells. You don't want material passing between cells. Certainly with your skin cells, you don't want bacteria or other pathogens slipping between cells. So tight junctions glue membranes together, typically close to the apical surface of the plasma membrane. We'll see other examples in a second. And there's proteins called occludin, claudin, and jam that do these. All right, so gap junctions allow communication. Tight junctions allow a barrier function. Neither one of them are strong because they're not necessarily linked to the cytoskeleton. For strength, you need cell adhesion molecules, CAMs. Okay, there are 4 different types of cell adhesion molecules that we're going to talk about, and they can be classified into 2 groups. There are homophilic cell adhesion molecules and heterophilic cell adhesion molecules. Homophilic, as the name suggests, it's the same molecule on both cells. So the cell has one family member, the other cell has the exact same protein, and those cells stick together because of those proteins. Heterophilic means that the proteins that are binding are different. So the cell adhesion molecule is binding to something else that may or may not be a cell adhesion molecule. So if you have a homophilic interaction, you know it's cell-cell. If it's homophilic, you know you have the same molecule on the other side, therefore it's cell-cell because it has to be. If it's heterophilic, you don't know if the interaction is cell-cell or cell something else. In this case, it's cell ECM. Okay, we'll go through those in a second. Hopefully it'll make more sense in a second. Okay, let's start off with these homophilic cell adhesion molecules. First of all, let's back up and say all of these cell adhesion molecules are transmembrane proteins. We'll talk about transmembrane proteins next lecture. Transmembrane proteins have an extracellular domain, part of it is outside the cell, has a transmembrane domain, and then has a cytoplasmic domain. So all of these homophilic or heterophilic cell adhesion molecules are transmembrane proteins. Importantly, they all have a cytoplasmic domain as well as an extracellular domain. The way these things are drawn here, it looks like they have these huge extracellular domains and these very small cytoplasmic domains. That isn't necessarily the case, that's just how they're drawn here. Okay, so the scale, the way things are drawn, ignores the artistic merit of this thing. All these molecules are transmembrane proteins. They have an extracellular domain, a transmembrane domain, and a cytoplasmic domain. Okay, so homophilic cell adhesion molecules. There's two types. Something called Ig superfamily of CAMs or cadherins. Okay, so let's look at those two. So these are homophilic, Ig superfamily, and cadherins. For the Ig superfamily, there's very little for you to memorize about them. They are called the Ig superfamily because they kind of look like immunoglobulins. Some of their domains looks like immunoglobulins. There's different types of these CAMs. So there's NCAM, ICAM, TCAM, PCAM. There's different family members. They're homophilic. So if one cell has one type of these molecules, the other cell has to have the same type for adhesion to occur. For example, if one cell is expressing NCAM, NCAM is neural cell adhesion molecule, the other cell has to have NCAM. That's why neural cells, for example, will stick together because they have the same homophilic cell adhesion molecule. So there's a variety of Ig superfamily CAMs, and they're homophilic, so if one cell has one type, the other cell has to have the same type. An important aspect of their function is that they're calcium independent. They don't need calcium to stick together. Okay, so that's the Ig superfamily of CAMs. The other molecules involved in homophilic cell adhesion are cadherins. Just like CAMs, there is a variety of different types of cadherins. There's probably more cadherins than CAMs. And again, they have different types, so different cells will have different types of cadherin. So there's E-cadherin, which is epidermal or epithelial. There's N-cadherin, which is neural cadherin. So same idea. If one cell has ECAD, the other cell has to have ECAD for them to stick together. So a reminder that these molecules are transmembrane proteins. They have an extracellular domain, a transmembrane domain, and a cytoplasmic domain. And it's these extracellular domains that recognize the proteins on the neighboring cell. So if one cell has ECAD, it's probably an epithelial cell. The other cell is going to have ECAD, it's also an epithelial cell. They can stick together. The same is true with the Ig superfamily. The difference being is that cadherins are calcium dependent. Cadherins need calcium for their extracellular domains to bind. But that's okay because we've already talked about there is no calcium in the cytoplasm, but there is tons of calcium extracellular. We have lots of calcium outside of our cells. There's always calcium outside of our cells. So in general, cadherins don't have a problem functioning in vivo, like in our body. Okay, so cadherin function isn't limited in the body because there's always extracellular calcium. But that fact, the fact that NCAMs are calcium independent and cadherins are calcium dependent, is a good tool for doing studies to try to figure out how these cells are stuck together. So if you have some cells in a dish and they're stuck together, if you remove the calcium, if they're still stuck together, they're probably held together by the Ig superfamily. If you remove the calcium and they fall apart, they're held together by cadherins because cadherins need calcium to function. Make sense? A reminder for both the cadherins and the Ig superfamily, they have extracellular domains that are involved in adhesion, but they have these cytoplasmic domains which aren't really drawn well here. Those cytoplasmic domains are important because they interact with the cytoskeleton. The reason that these molecules provide adhesion and strength is because they interact with the cytoskeleton. All right, so kind of looking at that with just the cadherin example here, but the Ig superfamily basically does the same thing. The cadherins can be linked to the cytoskeleton. Right, so here they're showing you two plasma membranes with a space in the middle. So there's a cell on top and a cell on the bottom. I always like to think of these as sort of epithelial cells, so if you want to just turn it on its side, you have an epithelial cell on the left and an epithelial cell on the right, and in between is the extracellular space. So in between these cells, you will have gap junctions. In between these cells, you will have tight junctions. And now in between these cells, you have these cell adhesion structures. Okay, so cadherins, transmembrane molecules, they have an extracellular domain that binds to the other cell. So these will all be the same cadherins. Okay, they have a transmembrane domain, and then they interact with the cytoskeleton. There's many proteins that link the cytoplasmic domain of the cadherin to the cytoskeleton. So here they're just called adaptive proteins. Different cells are going to have different proteins that link cadherin to the cytoskeleton. Okay, so we sort of talked about this without naming these things. So cadherins are forming cell-cell adhesions. These cadherins are going to be bounding to the extracellular domain outside of the cell, and then they have cytoplasmic domains on both sides. These cytoplasmic domains can interact with the cytoskeleton. If they interact with the intermediate filament cytoskeleton, that's called a desmosome. Okay, so a desmosome is cell-cell adhesion involving cadherin and intermediate filaments. I've already mentioned that before, but now we're kind of getting into more from the cadherin side of things. Okay, so it's homophilic, the cells are the same molecules, interacting with the intermediate filament cytoskeleton. In this case, the intermediate filament is keratin, so this is an epithelial cell. Okay. Cadherins can also come together to form adhesion proteins, and instead of linking to the intermediate filament cytoskeleton, they can link with the actin cytoskeleton. When they link to the actin cytoskeleton, they form something called adherens junctions. These adherens junctions can come together to form something called circumferential belts. So in lots of epithelial cells, you have these belts of actin that's contractile close to the apical surface. That belt is holding cells together. That belt involves cadherins. Okay, so there's two types of cell-cell adhesions that cadherin can play a role in. One is called desmosomes, and that's when it interacts with the intermediate filaments. And one is called adherens junction, and that's when it interacts with the actin cytoskeleton. Okay, for both of these interactions, the cadherins have a cytoplasmic domain, and there's a bunch of adapter proteins. So these adapter proteins allow the cell to know it's bound to another cell. So if you can imagine epithelial cells are supposed to form a barrier. If one cell is damaged or ripped apart, the rest of the cells need to know this because then you need to start a repair process. So a lot of the adapter proteins are actually signaling molecules. So that if you break this bond, if something is damaged with these cadherins, there's signaling cascades triggered that begin a repair process, right? So the cell knows if it's bound to another cell. Epithelial cells are happy when they're bound to epithelial cells. If they're not, there's signals that trigger repair processes, etc. For example, here's the protein beta-catenin that I'm sure a lot of you have heard of, that's involved in signaling. So if you damage these cadherins, beta-catenin will signal to the nucleus and start repair. Okay, so let's kind of start putting this together. Looking at sort of epithelial cells here, two epithelial cells that are drawn in a different way. So again, artistic license on how they're drawing things, why they're drawing things the way they are. It doesn't matter, but the logic of most of this should make sense. So you have two epithelial cells, an epithelial cell on the left, epithelial cell on the right. They have microvilli near the top. There's an apical surface. There's a basal surface here. This basal surface is linked to extracellular matrix, which we'll talk about in a second. So in between these epithelial cells are several types of things going on. There are gap junctions. They're drawn differently here, but regardless, there are gap junctions. So gap junctions are these connection proteins that form a channel. So these cells can pass ions back and forth between each other. In between these cells are tight junctions. So near the apical surface, the membranes of these cells are glued together to prevent things from going through. In between these cells are cadherins. Those cadherins can form desmosomes. So desmosomes are when the cadherins are linked to intermediate filaments. Here they've drawn the intermediate filaments in blue in some weird pattern. I'm not sure what the pattern represents. Regardless, there are desmosomes formed, and those desmosomes are linked to intermediate filaments. This is an epithelial cell, so that intermediate filament is keratin. So keratin is linking those desmosomes to other desmosomes and holding things together, giving it strength. Cadherins are also interacting with the actin cytoskeleton. Right? With the actin cytoskeleton, it's forming an adherens junction. That adherens junction is basically forming a belt around the top of the cell. So adherens junctions can form circumferential belts. Okay, so here you have gap junctions, tight junctions, and cadherins forming both desmosomes and adherens junctions. Okay, so those are all cell-cell, but what you also have here is cell ECM. So you have this thing called a hemidesmosome. That hemidesmosome is linking the cell to the extracellular matrix, providing strength by binding to the extracellular matrix. So this extracellular matrix, there's lots of different components of it, and I will just try to say extracellular matrix or ECM. Here there's basal lamina and connective tissue. These are all different types of extracellular matrix, but it's all ECM, extracellular matrix. So, what is extracellular matrix? A few things for you guys to memorize, things that it can do. I work on ECM, so you know it's important. Right? So, what was some functions of the ECM? So the ECM can anchor surrounding cells and hold things in three-dimensional space. I've mentioned already that as a multicellular organism, all your cells secrete ECM. So there's ECM surrounding all of your cells, and then all the cells hold on to that ECM. So all the cells are anchored to the ECM. The cells like being anchored to the ECM. Right? They get survival cues from that, and as I already mentioned with epithelial cells, the polarity. So when you look at the polarity of an epithelial cell, the basal surface is always ECM, the apical surface is always outside. So polarity is provided by ECM. They either allow or inhibit cell migration. I've already mentioned you need traction for cell migration. So if a cell is going to move, it has to move on the ECM or not. Because ECM is between cells, if cells are signaling to each other, the ECM can regulate what's being signaled. So growth factors that are passing between cells that can either be helped by the ECM or hindered by the ECM. So the ECM can allow signaling or inhibit signaling because it's actually between the cells where the molecules are going through it. Similarly, receptors are sitting on the cell surface facing the outside. Lots of receptors are facing the ECM. So not only can the ECM regulate the ligands that are floating around, but they can also regulate cell surface receptors. These receptors are literally sitting in the ECM. Okay, so it's these proteins that are outside of cells that can regulate lots of functions of the cells. There are dozens, if not hundreds, of proteins that make up the ECM. There's 3 broad classes that I'm going to mention and 4 proteins, I think, that you need to memorize. Okay, so ECM proteins can be hydrophilic. So they provide water pressure. So there are proteins called proteoglycans. I'm sure you guys have heard of these things: chondroitin sulfate proteoglycan, keratan sulfate proteoglycan. You can take little tablets. These proteoglycans are found in your joints, in your synovial fluid and things, but they're also found between cells. They're found in your skin. They absorb water. Okay, so hydrophilic means that they absorb water, and that absorption gives tissues resilience or lubrication, such as in your joints. But in your skin more so than my skin, a lot of the wrinkle freeness of it is because you have really nice proteoglycans, and as you get older, things start breaking down a little bit, and you have less hydrophilic molecules in there. Okay, so the ECM is hydrophilic. The ECM is structural. So there's large molecules that provide structure. The main one that likely everyone's heard of is collagen. There's many different types of collagen. Collagen can form many different structures. There's a lot of collagen in between your skin, in between your cells, I should say, that hold things together. But there's also large collagen structures. Your ears are made of keratin, your nose. So you can form large structures with collagen, or collagen can be in your tissues to provide support. Okay, so collagen is a large structural molecule. So proteoglycans are hydrophilic, collagen is structural. Then there is a number of adhesive molecules. So these are proteins that are present outside of cells to which cells can stick. Two of them are fibronectin and laminin. So there's little diagrams over here. You don't necessarily need to memorize any of the diagrams. Just appreciate what these proteins do. So either hydrophilic, structural, or involved in adhesion. And by adhesion, I mean cells can then move along. Okay, so fibronectin and laminin are adhesion molecules. It's not my fault, don't blame me, but don't confuse lamin and laminin. Lamin is in the nucleus, it's an intermediate filament. Laminin is an extracellular molecule involved in cell adhesion. I didn't do it, but the laminin outside the cell, lamin in the nucleus. All right, so you have the extracellular matrix. All cells are surrounded by extracellular matrix, and that sort of facilitates cell ECM adhesion. Okay, so now it's cell ECM adhesion. That involves a protein called integrins. Okay, integrins are heterophilic cell adhesion molecules. So integrins are binding something else, something different. Integrins are made up of a heterodimer. So there's an alpha and a beta integrin. I keep getting them backwards, but it doesn't really matter. There's several alphas and several betas. I think there's 20 alphas and 5 betas or something like that. So there's lots of alphas and beta combinations. And different combination of alpha and beta will bind to different things in the ECM. So what I would like you to memorize are these three. Alpha 1, beta 1 binds to collagen. Alpha 5, beta 1 binds to fibronectin, and alpha 6, beta 1 binds to laminin. If you remember when we talked about cell migration, we talked about filopodia protrusions and lamellipodia. So the membrane is protruded using actin polymerization. Then you need to form a new focal adhesion. That focal adhesion is made of integrins. So different cells will have different integrins. Different parts of your body have different ECM, especially in an embryo. So you can imagine in the embryo, you have muscle cells, you have nerve cells, you have sort of other endocrine cells or whatever. They're all migrating in different places. They're doing it by migrating on different ECM, so that the ECM provides a road. So muscle cells, you go this way, nerve cells, you go this way. But the cell has to have the right integrin to go along that road. So we talked about a cell protruding a filopodia and lamellipodia, and then it has to make a new focal adhesion. That focal adhesion is based on having the right integrin to bind to the right ECM. So if you have a dish, and you've coated that dish with fibronectin, and you put on a cell that has alpha 1, beta 1, that cell will not be able to migrate on that fibronectin because it cannot bind. Right? If that cell has alpha 5, beta 1, then it can move. So likewise in your body, the cell needs the right ECM to migrate. Right? So the integrins, this heterodimer of integrin has the specificity. However, interestingly, all integrins bind to the same core amino acid sequence. So in this example here, this is fibronectin. So fibronectin is a large molecule. Most of these ECM molecules are large molecule. They do many different things that we don't have time to go into. But almost all of these extracellular matrix molecules have in them somewhere a core RGD sequence. So those are the amino acids: arginine, glutamic acid, and aspartic acid. No, yes. No. Arginine, glycine, aspartic acid. Yes, thank you. Okay, RGD you have to memorize. Okay, so all integrins recognize the amino acids RGD at the core. However, there are other synergy regions around that RGD. So fibronectin will have RGD, but around that RGD sequence are other sequences that's recognized by alpha 5 beta 1. So alpha 5 beta 1 recognizes the RGD in fibronectin, and then it recognizes other amino acid sequences around that tells it this is fibronectin, it's okay. Alpha 1 beta 1 recognizes RGD in collagen, but it recognizes the other sequences around that to tell it this is collagen. In terms of laminin, it recognizes the RGD in laminin, but it tells it there's other sequences around the laminin that it also recognizes. So they're specific. At the core, they're all binding to RGD. However, there's other sequences around it that give them specificity. All right, so integrins are transmembrane molecules. We mentioned that before. I mentioned at the beginning of this lecture as well. So integrins have an extracellular domain, integrins have a transmembrane domain, and integrins have a cytoplasmic domain. They've drawn them different here. Again, the artwork doesn't really matter. Part of integrin is extracellular, part of it is transmembrane, part of it is cytoplasmic. Okay, on the extracellular side, integrin is going to bind to the extracellular matrix. So here you have an example of binding to fibronectin or laminin. So extracellularly, it's going to bind to the extracellular matrix. Intracellularly, it can bind to the cytoskeleton. And just like cadherins, it can either bind to intermediate filaments or it can bind to actin. We already sort of saw this when we talked about focal adhesions. Focal adhesions are formed by migrating cells. So a focal adhesion is protruding a membrane, it has integrins in it. If it finds the right ECM molecule, it makes a focal adhesion. In this example here, this is fibronectin. So this integrin is alpha 5 beta 1. It forms a focal adhesion. Then from that focal adhesion, it forms a stress fiber, which is actin. So focal adhesions involve an adhesion with actin and extracellular matrix. And that's a stress fiber. Integrins can also be present in non-migratory cells. So in epithelial cells, they form these structures called hemidesmosomes. So hemidesmosomes are involved in integrin extracellular domain binding to the ECM. In this case, ECM is binding to the laminin, so this is alpha 6 beta 1. And then on the cytoplasmic side, it's binding to intermediate filaments. So a hemidesmosome is bound to intermediate filaments, a focal adhesion is bound to actin filaments. Okay? However, focal adhesions are formed with integrin and actin. Focal adhesions are found in migrating cells. Hemidesmosomes are formed between integrin and intermediate filaments. Hemidesmosomes are almost always found in non-migratory cells, such as epithelial cells. So the way they've drawn this, where it looks like there's one plasma membrane, where it looks like it has both hemidesmosomes and focal adhesion, that's not likely. A cell can't both be standing still and moving, so it's not likely you'll find a hemidesmosome and a focal adhesion. This intermediate filament is keratin. Keratin further suggests it's an epithelial cell. Again, epithelial cell wouldn't be migrating, so an epithelial cell wouldn't have stress fibers. So this is just a bad diagram. But let me just back up to this then. All right, so you have cell adhesions. So you have gap junctions, tight junctions, desmosomes, which are cadherin intermediate filament, and adherens junctions, which are cadherins and actin. You also have adhesions to the extracellular matrix. In the case of an epithelial cell, this is a hemidesmosome. You're binding the extracellular matrix through integrins to the intermediate filaments. If this was a migrating cell, you would have a focal adhesion instead of a hemidesmosome, because you're binding to the actin cytoskeleton. Okay, it'll all come together in a table in a little bit, hopefully. All right, so the other thing about integrins is they can be in an active or an inactive form. So there's a lot of stuff here that's showing you lots of things. The most important part is at the top. Integrins can be activated or inactivated, and the cell regulates what's happening. And you can imagine this is necessary during cell migration. So during cell migration, the cell might have integrins on it, but you don't want those integrins activated until the cell is ready to move. So if you have a cell that's not really doing anything, you don't want the integrins activated. Once you polymerize actin, once you make filopodia and lamellipodia, then you want the actin to be polymerized. So you want the integrin to be activated. And you can ignore most of this, just the logic of it. You don't have to memorize this, I won't test you on this, but you can see that CDC42, RAC, and Rho are involved in actin, so there's signaling back and forth. So when the cell is beginning to migrate, one of the things it needs to do is activate integrins, which makes sense. Okay, so that's all you need from this, is that integrins can be in an active or an inactive form. Okay, so we had two homophilic cell adhesion molecules. We had the NCAM, the IG superfamily of NCAM, the cadherins. Heterophilic, we just talked about integrins. And now, heterophilic, we're also going to talk about these molecules called selectins. And we're going to use this example of extravasation, which actually includes a lot of these different types of cell adhesions to kind of kind of tell you what's going on here. Okay, so selectins recognize sugars, oligosaccharides. So all of your cells have proteins on the cell surface. Most of those proteins are glycosylated in certain ways. So your cells have not only sugars on them, but a lot of the proteins are glycosylated. So there's all kinds of oligosaccharides on different cells, and different cells have different sugars on them. Selectins are a family of cell surface molecules that bind to sugars. So there's different types of selectins that can recognize different types of sugar. In this example, there is a selectin called p-selectin that's going to recognize a specific type of sugar. And the process we're going to look at is something called extravasation. So I'm going to go through this. We're going to watch a movie, and then we're going to come back to this. So just to kind of remind you though first, okay? So we talked about integrins, which are transmembrane molecules, and now we're talking about selectins. So selectins are transmembrane molecules, and they're going to bind to sugars. You don't know where those sugars are. Those sugars could be from another cell, or those sugars could be from extracellular matrix, for example. So you don't know the type of adhesion. That's why these guys are heterophilic. You don't know what they're going to bind to, but it's certainly heterophilic. They're not binding to other molecules like themselves. All right, extravasation. What do we got? We have endothelial cells. So endothelial cells are a modified epithelial cell that lines your bloodstream. So your bloodstream has cells that are tightly bound to each other. Okay, so your bloodstream endothelial cells have molecules in between them, including cell adhesion molecules, including one called ICAM. So even though it's not drawn here, in between these endothelial cells is ICAM molecules. There are other ICAM molecules that are not in between them, and the way they've drawn molecules here is huge. I'm not sure why they drew them that big, but obviously molecules aren't that big compared to the cell. This is for artistic license. So these endothelial cells are bound together using cell adhesion molecules. Perfect. They probably have gap junctions between them as well, probably tight junctions too. So you have these cells that are tightly bound. On one side of the cell, basically the outside, is where you have your bloodstream. In your bloodstream, you have a variety of immune cells, including leukocytes. The job of leukocytes is to go fight infection. So if you prick yourself with a dirty needle or something, you're going to have some sort of pathogen somewhere in your tissues. In principle, the leukocytes have to leave the bloodstream and get out into the tissue to fight this pathogen. So extravasation is when the leukocyte leaves the bloodstream and goes to fight the invasion. So you have all kinds of blood cells, all kinds of things streaming through your bloodstream. You do not want anything sticking to the endothelial cells. That's a bad thing. Once you start getting plaques and growths on the endothelial cells, that's not good. So in general, you do not want any cell adhesion between cells in your bloodstream and the endothelial cells. You just want them to keep on going. However, in the case of this sort of immune response, you do want the leukocyte to bind to and then leave the bloodstream. So here the leukocyte has to bind to the endothelial cell in order to go outside the endothelial cell barrier to fight the pathogen. So far making sense? So how does this work? Well, when you have a pathogen, that pathogen is going to trigger a bit of an immune response. So one of the things that immune response does is it causes the secretion of a selectin on the endothelial cells. So if there is no pathogen, there is no selectin on the endothelial cell membrane. In the presence of the pathogen, you will get P-selectin is secreted to the plasma membrane. So in the response to a pathogen, P-selectin is secreted to the plasma membrane of an endothelial cell close to where the pathogen is. Make sense so far? P-selectin bonds to carbohydrates. The carbohydrates it binds to happen to be on the leukocyte, on the specific leukocyte. Okay, so not other blood cells or other things, just this leukocyte that's going to be recruited. So the P-selectin will then bind to the sugars that are found on the leukocyte and basically bring the leukocyte close to the endothelial cell. So P-selectin binds to the sugars on the cell surface of the leukocyte and brings that leukocyte close to the cell surface. The endothelial cell has something called PAC, and the leukocyte has something called the PAC receptor. PAC is platelet activating factor, but it doesn't really matter. Okay, so the endothelial cell has PAC, the leukocyte has the PAC receptor. Here, P-selectin brings them together, and PAC binds to the PAC receptor. When PAC binds to the PAC receptor, it activates integrin. There's an integrin that's present on the leukocyte. Prior to this, so without PAC binding to the PAC receptor, the integrin is not active, but when PAC binds to the PAC receptor, the integrin is active. Easy? When that integrin is active, it can now bind to its substrate. This integrin is a weird one, but you don't need to memorize it. It's somewhere here. Alpha L beta 2. You don't need to memorize it, but it's a specific integrin and it's specific for something. So this integrin binds to a protein that has an RGD sequence in it, and then it recognizes other sequences around that RGD. Other proteins, in addition to extracellular matrix, can have such sequences, such RGD sequences. And in this case, the cell adhesion molecule that these endothelial cells have has that RGD. So this molecule here called ICAM. So this ICAM is present and is involved in cell adhesion, but that ICAM also has the RGD sequence and the synergy sequences that's recognized by this integrin that's on the leukocyte. So when the integrin is activated, it binds to this ICAM molecule. And this binding of the integrin to the ICAM molecule is strong. It's very powerful. It stops the cell, and it actually allows the cell to bind tightly to the endothelial cells, and then the leukocyte can squeeze in between the endothelial cells and go off and fight the infection. So ultimately, what you want is a really strong adhesion, and that's the ICAM molecule being bound to by integrin, but the integrin has to be activated. So it's a cascade of increasing cell adhesion ending with integrin binding to ICAM. So it's starting off with P-selectin being secreted in response to the pathogen. The P-selectin is going to bind to sugar on the leukocyte. So that's one type of heterophilic adhesion. Then PAX is going to bind to this PAX receptor. That's going to activate integrin, and once that's activated, that allows another heterophilic adhesion in between the integrin and its ICAM molecule. The ICAM molecule is also present undergoing cell-cell adhesion, that's homophilic, but in addition to it doing homophilic cell adhesion, it's now playing a role in heterophilic cell adhesion as well. Okay, I'll show the movie next time. It should be online. Hopefully you guys can watch it all by yourself. See you guys Wednesday. Turn the mic off.
Is the binding that I cascade that's like ripping apart the gal junctions?
Yes, yes. Yes, it's not damaging